affecting our respiratory tract, and it's caused by a virus known as severe acute respiratory syndrome type 2. In short, it's SARS-CoV-2. More or less, it's like a SARS, but it's signified with a 2 lah. SARS is 1. And it has induced a global pandemic that is responsible for over 6 million deaths for the last 3 years, 2021, 22, right? Oh, now we are in 23. Deaths are still piling anyway. Okay, this SARS-CoV-2 virus or COVID-19 virus enters and infects our body through the binding of its spike protein to the receptor protein known as angiotensin converting enzyme 2, in short ACE2, present on the surface of the target cells in our body. All right, although vaccines have been developed to prevent this viral infection, but due to uneven global vaccination and high transmission rate in some countries, the virus has mutated, right, to an extent that is beyond the efficacy of the original vaccine. So now vaccine is not that effective anymore, not like originally they say, oh, 95%, 80% effective. Now it's like only maybe 60%. People who are vaccinated are still infected, right? Okay, so to develop a new vaccine, it takes time, all right, and may not keep up with the new viral mutations. I have been mutating now. Now it's a what XXBB or something like that. Therefore, an alternative strategies that are safe and non-toxic are needed to reduce the rate of this viral infection and to tackle the constantly mutating virus, right? Okay, next one, next page. So in 2022, a joint research by the Institute of Traditional Medicine and the Institute of Microbiology and Immunology of Yangming University in Taiwan, Yangming Chiaotong University in Taiwan, they found that immunomodulatory protein cloned from Ganoderma microsporum can make the ACE2 receptor on the surface of the cells disappear, and yet not damaging to the cells. So that means it's non-toxic lah, thus reducing the chances of the viral spike protein binding to the target cells that cause infection. Okay, the study led by Xing Ye was published in the journal Phytomedicine in 2022, titled GMI, a protein from Ganoderma microsporum induces ACE2 degradation to elevate infection of SARS-CoV-2 spike pseudotype virus. So those who want to read more of the research, you can type this title, you can have a free access to it. Okay? All right. Okay, now talk a little bit about ACE2. Right, ACE2, angiotensin converting enzyme 2, is a protein found on the surface of many cell types and tissue in our lungs, in our heart, in our blood vessel, in our kidneys, in our liver, and in our gastrointestinal tract. Okay, it is a, later I will have a diagram to show you about it. The spike protein of the SARS-CoV-2 virus binds, that means it connects to this ACE2 to enter and infect the cells, causing the infection known as COVID-19. So hence ACE2 is also considered a receptor lah. ACE2 is also a receptor. When you talk about receptor, that means something that stick to it is called a receptor. Okay, this ACE2 ACE2 actually is a modulating enzyme. It modulates the blood pressure, inflammation, and prevent damages to the blood vessel linings in our blood vessel, in the kidney, here and there. All right. This is the normal function of the ACE2. Okay, this is the normal function. But unfortunately, this SARS-CoV-2 virus spike protein binds to it. So, when this SARS-CoV-2 virus binds to it, then ACE2 will be prevented from performing its normal function, right? So, potentially causing increased inflammation and death to cells in the lungs. That's why people who suffer from COVID-19 say that they are short of breath. They have cytokine bomb or something like that, inflammation bomb or something like that. This is the reason because they bind to it. This is a regulator. So ACE2 inhibitors, as we all know, that is commonly prescribed to people with hypertension, high blood pressure. This is only inhibit the enzyme ACE1. Okay, ACE1 and ACE2 is like our T helper cell 1, T helper cell 2. If you understand about this T helper cell 1, T helper cell 2, they balance one another. So the ACE inhibitors only inhibit the ACE1, cannot inhibit this ACE2. So ACE inhibitors is not effective as a control of the COVID-19 virus. However, this angiotensin ARB blocks ACE2. I didn't quite, I forget what's the full name. Can be considered. This is also another medication that is used for people with blood pressure problem. So this one has been considered. So that's why in some like the vaccination medication is this ARB blocks. All right. Okay, but anyway, just an idea lah. I mean, if you want to know more about it, then you could read on it. Okay. Next one. Okay, let's look a little bit about the materials and the methods. Okay, Ganoderma immunomodulatory protein is a fungal immunomodulatory protein cloned from the species of Lingzhi known as Ganoderma microsporum. This Ganoderma microsporum is found in Taiwan. Actually, Ganoderma immunomodulatory protein is what we normally know Lingzhi 8, LZ8. We sometimes in our this the what 过图 we always talk about LZ8, right? LZ8 is one of the immunomodulatory protein. So it's the same lah. So you can also find this in normal Lingzhi. Right. So research has shown that this Ganoderma immunomodulatory protein has anti-cancer activities, especially against lung cancer, as it triggers protein degradation of the pathway. When degradation, that means they show that reduction. Degradation means a reduction, resulting in the death of cancer cells. So this suggests that this immunomodulatory protein may target lung cells and induce protein degradation. Therefore, that's why they use this for the study. This is the push behind why they're going to pick this and not pick others. Because there have been a research on this to show that it has affected the lungs. Right? So the effects of GMI in short, because when you talk about Ganoderma immunomodulatory protein, the tongue keep on twisting, I also find a problem. Right? The effects of GMI were first examined in vitro. Right? This is the method. Okay, how they do? They first do an in vitro study, right, to compare ACE2 present in this HEK293 cells. This HEK293 cells is a cell that is found in our kidneys. All right. Why they pick this? Because this is a cell that is normally used to study virus, because they have this binding effect, right? It's exposed. So, like I say here, HEK293 T cell is a derivative human cell line and are commonly used in research on viral infection and vaccine development. All right. And then they cultivate a SARS-CoV-2 pseudovirus. Right? This pseudovirus is not exactly the virus of the COVID-2, but they did use COVID-19 virus and implant it into some plasmid, and then somehow or other, some measure that they go about to do it, that end up become a virus, kind of similar to a SARS-CoV-2. So that's why they call it pseudovirus, not the true virus. I have to qualify this, all right? Qualify this, right? Okay, then after that, they did an animal study on the inhalation of GMI to see whether it's effective or not. And this inhalation is through a nebulizer to see the effect. All right, why we want to adopt this inhalation method is because COVID-19 that generally infect us through inhaling, okay, through our respiratory tract. We breathe in, it affects our lungs. That's why the study adopt this method. Okay, next. All right, so they did that. The study, what's the result? What is the result of the in vitro? In vitro, that means study just on the dish. Some cell put on the dish, not on the body somehow, just the cell movement. Right? They found that GMI doesn't affect the viability of HEK293T and the ACE2 receptor, all right, in the 24-hour to 72-hour of treatment that they have done. That means the cell still survives, still around. So that means it's not toxic. It doesn't mean that you apply the GMI on it, the cells all die out. Then it's not viable. So when you say not affecting the cell viability, that means the cell is still surviving lah. So they use a dosage of 0.06 micrometer. GMI effectively, they say effectively reduce the SARS-CoV pseudovirus infection rate by up to 80%. So you can see the diagram on the right, the extreme right, the chart. Okay, see the control where it is just using the saline, rubbing on the the cell. It doesn't have any effect. But if they use different dosage, the number of cell infection fell from 60% to only 20%, 20 of that. So that means it can reduce by 80%. Oh yeah, I run out of power again. Hey, okay, so and then they also found that this GMI reduced SARS-CoV-2 pseudovirus infection of the epithelial cells in the lung. I mean the lining in the lung cell, the alveoli, those the sac in the lungs. Okay, that change from the air we breathe in into the oxygen our body need, right? So this finding suggests that GMI can reduce ACE receptor expression and may act as a potential agent to inhibit. All right. So the in vitro outside the cell on a dish experiment show that it is feasible. They can inhibit SARS-CoV-2 infection. So therefore, the next step they go on, they do an animal study. All right. Okay, next one. Okay, they do the animal study. Right. Remember, I talked about the nebulizer. They put the rats into the box. Right. They have a nebulizer. How they do it? They mix 100 microgram of GMI with 1 milliliter of saline, then they atomize it and put in a nebulizer and spray it at a rate of I think 0.25 per, per minute, such that it's like they only expose for 30 seconds, 30 minutes one time. Then after that, they put the mice inside this box and they spray the GMI that has been nebulized on it to let the rats breathe. Okay, 30 minutes later, they stop. Then 6 hours later, they kill the mice and extract the lung tissue to study. All right, then they do again another group. 24 hours later, they do the same thing. They spray and expose for half an hour. Then after that, they kill and extract the lung. All right, and measure the ACE2 in this lung tissue. All right, the results show that regardless of whether the mice inhale only one time or 24 hours later inhale again, the ACE2 level in the lung tissue of the GMI group was 60% lower than the control group. All right. So what does this tell you? This shows that the dosage equivalent to 1.125 grams of GMI per kilo of body weight is enough to protect the lungs for a whole day. Okay, so it reduces this ACI receptor level in the lung. Okay, just remember I tell you the ACI receptor in the lung, when the ACI receptor is there, the virus can bind. So when it reduces, the virus cannot bind. So in the sense, in this way is one of the preventions. All right. Next level. Okay, what is the mechanism of action? Next slide. All right. You look at the study found out that the reduced level of sites was due to the endocytosis. That means the degradation or these ACE2 cells were being absorbed into the cell. All right, eaten by the cells itself, by this process called proteasome and lysosome degradation. That means GMI will activate this endocytosis of ACE2. That means reduce it. You see the brown color Y that you see on the big cell. There's a whole cell. That is a receptor ACE2. So the small circle you see is the virus, right? The virus has all the spike that's supposed to stick to this to enter the body, all right? So once this Y thing that you see doesn't exist, it cannot bind, all right? Then at the same time, it telling you the study found that this reduction of ACE2 was only temporary, right? and only affect 20%. Remember just now I was telling you ACE2 has an important function for our blood pressure, but as it is, although GMI reduces ACE2 level, but it doesn't completely reduce it. All right, you only reduce by about 20%, and at the same time, it absorbs into the cell, host cell, to be recycled. Okay, the recovery, of course, when you stop consuming GMI, the ACE2 will recover, but a bit slower lah, huh? After 2 days, it's still less than 50% has recovered. All right, so what this research here tell you? It tell you that GMI has denied an entry point for the virus. That means no more ACE2, right, to the host cell. And this denial can last a longer period, all right? And at the same time, it doesn't affect the regulatory function of ACE2 because you only affect 20%. And you only do it so when it is necessary. So what is necessary? When there's a viral infection, you consume it, you reduce it. All right? Okay. So, okay, next slide. So in addition, the study also found that GMI binds to the ST domain, S2 domain of the spike protein. You know the spike protein, if you look behind just now, the spike protein, the black purple dot is GMI. GMI stick to the spike protein, the spike on the virus. All right. So by right, the spike on the virus supposed to lock into the ACE2, but because GMI stick to it, it cannot lock in. It cannot lock into the ACE2. So as a result, it failed to lock in. That's exactly the second mechanism. But this second mechanism is not very active. It's only 1/3. All right, it's only 1/3 of the previous mechanism of reducing ACE2. Okay, but of course, this is also one that can help. All right, anything that can help to reduce infection plays a part. So it has two mechanisms here. So this is one of the secondary mechanism. All right, the primary mechanism is that GMI reduce ACE2. The secondary mechanism is that GMI binds to the spike protein of the virus. So as a result, the virus cannot bind to ACE2. Okay, right. So what is the conclusion? The conclusion is that as ACE2 is a gateway to the virus, right, to enter the body. So when you stop it, even the virus mutate to any kind of thing, it doesn't matter because there's no ACE2 for them to bind to, right? So therefore, when they cannot bind to, there cannot be an infection. So whatever the mutation is not to worry, right? So this study plus two other studies that we have previously shown here is that Lingzhi polysaccharide can inhibit the growth of COVID-19 virus through intestinal immunity. That was done, I think, a few months ago. Then there was also a computer selection of Lingzhi triptanoid that show that 4 types of Lingzhi triptanoid can inhibit COVID-19 virus infection. So all this shows that Lingzhi is basically a godsend herb to help us tackle the SARS-CoV-2 virus and pandemic. It therefore will be a disappointment if we did not take advantage of it. All right. Okay, that's all my sharing. Thank you very much. Thank you. Any question?
Okay, thank you.